So, hello. Welcome, everybody. It's a pleasure for me to share with you reflections about the new data of human tuberculosis in prehistoric period. In the beginning, I would like to thank the association Arwa for giving me this chance to present my work through this window. I will review with you the archaeological evidence of human tuberculosis during the Neolithic period coming from the Fertile Crescent. Actually, the study of history and evolution of infection disease is a topical subject to increase our knowledge about the emergence of infections and epidemics. It's also a subject in a development for physical anthropology and paleopathology. This approach can be defined as the science of diseases on human and animal in the past. The first purpose of our program is to investigate the paleopathological evidence of tuberculosis before domestication process. I start this approach during my PhD by a pluridisciplinary analysis carried on human bones from 2 Neolithic sites, representing pre- and early domestication phases in ancient Syria. Afterward, in order to shed further light on the debate concerning the origin and evolution pathway of tuberculosis, I include actually during my postdoctoral new Paleolithic and Neolithic sites from Turkey, Syria, and Jordan. In the beginning, tuberculosis is an infection disease caused by germ of mycobacteria, called Mycobacterium tuberculosis. It spreads from person to person through the air and usually affects the lungs. Generally, there are 2 forms to develop the disease. In the case of latent tuberculosis, you have the germ in your body, but your immune system stops them from spreading. You don't have any symptoms and you are not contagious, but the infection accidentally can become active. The second form for tuberculosis is active tuberculosis. This means the germ multiply and you are contagious. In this case, bacteria can affect other parts of the body, such as the spine, articulation, and system of long bone. The family of Mycobacterium tuberculosis complex comprise other members of Mycobacteria and affect human and many animal species. The human tuberculosis strain include 4 members: Mycobacterium tuberculosis, Africanus, Microti, and Caniti. However, the cattle tuberculosis strain of bovis include only Mycobacterium bovis. Until very recently, the widely accepted hypothesis about the emergence of tuberculosis suggests the human pathogen derived from cattle by contamination during the Neolithic, and following a model of bacterial speciation. According to this scenario, the bovine tuberculosis strain was considered the precursor of Mycobacterium tuberculosis human. This hypothesis was based in particular on the absence of paleopathological case identified on human population before the Neolithic, its presence attested only on animal skeleton. This paleopathological consensus matches with the oldest evidence of human tuberculosis, identified only in the cradle of cattle domestication. The Neolithic is the interesting period concerned by our research. During that, numerous factors should be considered in favoring the emergence and spreading of pathogen. On one hand, a bacterial species jump from animal to human due to proximity with animal, and during this phase of the first phases of domestication. On the other hand, the dramatic increase of population density in the Neolithic order after the sedentarization. During this period, we have the strongly suggesting cases of tuberculosis were observed at the Neolithic site from Middle East and showing evidence of agriculture and animal domestication. The first one, the site of Ain Ghazal in Jordan, providing 3 evidence of human tuberculosis discovered in the level of the pre-pottery Neolithic sea. From the same period, the site of Atletiam presented 2 evidence and confirmed by lipid biomarkers and ancient DNA. So, for the Paleolithic period, evidence of tuberculosis is poorly documented. It was only attested on skeleton of bison. The paleopathological legion on bison remains observed in the natural trap cave in Wyoming and dated from 17,000 before present. For humans, the oldest case of tuberculosis would have been reported on fragmentary skull of Homo erectus, discovered in Kocabaş in Turkey. The endocranial surface exhibits some changes that have been interpreted by Kappelman and cohorts as possibly resulting from tuberculosis of meninges. However, some critical points reported by other paleopathologists, and this case no more debated afterward. All previous paleopathological evidence attested that tuberculosis appeared in the Neolithic. However, the studies of molecular phylogeny of the Mycobacterium tuberculosis has challenged this dogma and provided a new evolutionary scenario, suggesting that all members of Mycobacterium tuberculosis, including bovis, actually represent a direct descendants of a most ancient human strain. According with this result, the tubercle bacilli could have been already human pathogen before animal domestication. As a consequence, human tuberculosis cannot derive from cattle, but a contrary. The new molecular phylogeny estimate also the age of common ancestor of Mycobacterium tuberculosis about 3 million years old, and suggest also that human pathogen moved with Homo sapiens during the out of Africa. Following this scenario, we believe the paleopathology should be able to highlight human tuberculosis evidence prior to the process of animal domestication and coming from Paleolithic or Mesolithic period. On one hand, because the tuberculosis in one of the diseases that leave visible traces on bone. On the other hand, the methodological progress made it possible to increase the number of identified cases from archaeological context. Resulting from our disciplinary analysis, we reported strong evidence of tuberculosis. The osteoarchaeological material provided from two Neolithic sites from 9th millennium BC, the site of Dja'de el-Mughara on the left bank of the Euphrates, and Tel Aswad located in the central Levant near Damascus. Both sites are dating to earlier phases of the Neolithization process and corresponding to pre- and early animal domestication during the so-called pre-Pottery Neolithic P. Dja'de el-Mughara presents a very interesting archaeological context. In the early phase Dja'de 1 and Dja'de 2, no morphological evidence of plant or animal were domesticated. But during the third period, Dja'de 3, the site shows some evidence of agricultural, economic, and pre-domestication of Arabs. This site delivered also the skeletal remains of 130 individuals coming from different funeral practice and dating at the 9th millennium BC. Coming from period the end of PPNA and the early PPNB. The second site is Tel Aswad. The domesticated animal and plant resource has been exploited during the earliest occupation levels from middle PPNE and early PPNB, and hunting progressively reduced in this site. From Tel Aswad, skeletal remains of 119 individuals were discovered. So, in order to apply our paleopathological approach, we performed a macro-morphological analysis on skeletal alteration related to tuberculosis. Their diagnosis was based on the morphological aspect and the distribution of lesion on skeleton according to the criteria accepted in paleopathology. Afterward, interesting bone with paleopathological lesion were the subject of further paleoradiology and paleobiochemical analysis of lipid biomarkers or ancient DNA. In paleopathology, the bone changes due to tuberculosis can be classified into 4 groups. The first one concerns the typical lesion, such as spondylolisthesis tuberculosis or bot disease. This is a destructive lesion for 1 to 4 vertebrae, generally forming by structural kyphos of the spine. The second group comprises 2 types of lesion, diaphyseal lesion on the long or tubular bone, due to different forms of osteomyelitis, such as spina ventosa. And the second type of osteoarthritis tuberculosis is the joint involvement, such as spine, hip, and knee. The third group concerns the periostal reaction, characterized by new bone formation, symmetrical, single or multiple layers, observed on all long bone and tubular bones. This aspect corresponding with hypertrophic pulmonary osteopathy linked with intrathoracic bone disease and tuberculosis can one of the etiological proposed. The fourth group comprises no specific lesion or minor lesion. Among that, we have the periostal reaction on the internal surface of the ribs. Also, the endocrinial lesion due to tuberculosis leptomeningitis. This one called serpens endocrinia symmetrica. In this group, we can classify the superficial spinal lesion on the anterior part of the vertebral body. This type of vertebral lesion, described in the end of 19th century as a form of spinal tuberculosis, distinct other than the pod disease, and characterized by periosteal reaction developed around the enlarged plexus foramina. Our paleopathological study gave evidence of 10 tuberculosis cases coming from the phase PPNB. 3 tuberculosis cases observed in the earliest phase, Dja'de 2. And during this phase, we have no animal domesticated. And dating for the first half of 9th millennium. All cases characterized by visible layers of diffuse periosteal reaction on long bone and on the ribs, associated with superficial spinal changes on thoracic and lumbar vertebrae. These layers of new bone formation matches with criteria of hypertrophic pulmonary osteopathy caused by mycobacteria. In this collection, the third case of immature provide a positive result of biochemical analysis of lipid biomarkers made by Professor David Minikin from the School of Bioscience from Birmingham University. The profile of mucoleic acid and mucolipinic acid obtained correspond and similar with the profile standard of Mycobacterium tuberculosis. The same individual show also a positive result of ancient DNA, 2 sequence of the specific region of TPE7110 obtained by PCR. This analysis carried by Professor Albert Zenk and Frank Mixner from ORAC lab from Bolzano. In the same site of Dja'de el-Mughara, we observed 7 other cases from the recent phase Dja'de 3, dating at the second half of 9th millennium BC. During this period, we have the cattle domestication were described. 2 types of lesion were observed on bones, typical aspect of pod disease on adults, and superficial lesion with layers of new bone formation on 6 immature. For the first case, the present superficial vertebral change and the 3D reconstruction evidenced clear focal microarchitectural changes and match with early stage of infection. This micro-CT and 3D reconstruction applied by Helen Coconio from Bordeaux University. Among the material of Tal Aswad, 2 cases was observed. The first one present 2 types of lesion. Layers of new bone formation on all long bone on the hand and foot, corresponding with criteria of hypertrophic pulmonary osteopathy. And on the right ulna, we are a clear lesion of osteomyelitis, known in pathology by spina ventosa. The paleoradiology of micro-CT on the right ulna confirm our diagnosis of osteomyelitis and showing the clear lesion of spina ventosa. The second case from Tal Aswad concern a skeletal remnants from a child from early PPNB horizon. This case showing a legion of serpents endocrinia symmetrica on the internal part of skull attributed to leptomeningitis tuberculosis. We observed also a superficial lesion on the anterior part of 4 thoracic vertebrae. Resulting from our data, we don't have a strong evidence for the Paleolithic period. However, for the Neolithic, the number of paleopathological cases of tuberculosis increased on the Fertile Crescent. A new osteoarchaeological material were examined from Muraybat and Sheikh Hassan from Syria and Ain Ghazal from Jordan. We plan a new paleopathological analysis for Paleolithic site from Turkey and Syria. During this program, method of paleomicrobiology lipid biomarkers or ancient DNA are applicable only on the Neolithic sample. We proposed method of paleoradiology by micro-CT that offers the interest to be no destructive for the ancient sample and that's been successfully used in paleopathology. Animal bone coming from the site of Dja'de el-Mughara were examined and sampled for molecular and synchrotron analysis. However, no paleopathological evidence were observed. Following our data, we can conclude 3 old cases identified before domestication among the 10 cases from Neolithic period of Dja'de and Tel Aswad. We propose also 17 unpublished and new cases recently identified from Muraybat, Sheikh Hassan, and Ain Ghazal. For the Paleolithic period, we have only one possible case, but need to be confirmed by preliminary study at Kojabash fossil. Our paleopathological evidence predating or accompanying the animal domestication support the new model of human tuberculosis ancestral origin, and we do not exclude that tuberculosis bacilli were already circulating in Paleolithic population and spread with sedentarization. Actually, the few number of tuberculosis cases from the Paleolithic support this model, because the human density has been increased following the sedentarization. During the early phase of during the earliest phase of Neolithization, allow to reactivation and expansion of disease on human group. Finally, I would like to thank my collaborator during this last years and all archaeologists for providing us the access for the osteoarchaeological material. And then I would like to thank the association Arwa for this invitation. And thank you for your attention. Please let me know if you wish any question or any article about this subject. Thank you.